Hello, everyone. My name is Amy Zheng. I'm a PhD student at Vanderbilt University, and today I will be discussing my research, which is modeling carbon metabolism in the diatom, Phaeodactylum, and tricornutum due to genetic knockout of the tag degradation enzymes and due to changes in light intensity. So for the background and objective, I have two objectives for this project. One is to be able to connect central carbon metabolism to photosynthetic activity, and two is to optimize tag production using 13C metabolic flux analysis. So an overview of metabolic flux analysis is that it combines experimental isotope labeling studies along with metabolic models in order to describe the metabolism of the diatom under different conditions. And there are two different conditions that are discussed in this poster presentation. One is changes in metabolism due to light intensity, and then also changes in metabolism due to comparing the wild type versus an ACAD knockout, so acetyl-CoA dehydrogenase knockout, which prevents tag degradation. So the experimental procedure for isotope labeling study is to add a tracer into the diatom culture, which in our case is labeled sodium bicarbonate, and you measure the enrichment over time. And then you can compare that to a network model. So this network model contains all of the reactions that we are interested in. And by combining the network model with the experimental data, we can get a picture of what is happening with all of the reactions that we're interested in that are included in central carbon metabolism and how that relates to the greater organism. So this is done through an iterative process. When our simulated results from our network model match our experimental data, that's when we can get our flux map. So in figure 2 here is an example of a flux map, and this is what I've been using for Phaeodactylum tricornutum. This is based on the enzyme notations and literature data on the metabolism of central carbon metabolism in the diatom. So in the chloroplast, we have the Calvin cycle, and the TCA cycle occurs in the mitochondria. We also have two extra pathways. One is triacylaminerin synthesis, and the other is tag synthesis, which is connected through triose phosphate and acetyl-CoA. So the reason why we use central carbon metabolism is because the tracer, which is our case CO2, can be incorporated quickly. And our prediction is that the bottleneck for tag degradation and changes in photosynthetic activity can be seen in central carbon metabolism. So how you can read a flux map is the wider the arrow, the more flux is going through that reaction. And by using this flux map, ideally we would be able to predict bottlenecks in our system and changes in our metabolism of the diatom under different conditions. And there are two sets of instruments that we use to measure these metabolites. One is the GCMS, which we typically get TCA cycle and amino acid, and the LCMS, which is consists of the Calvin cycle metabolites. So going into the two different conditions, first let's talk about the light intensities. So we have two different light intensities that we're testing. The low light is the 60 microeinstein case, and the high light is the 250. So average percent enrichment tells us how much of the labeling has been incorporated into the metabolite. So we can see that at the beginning, comparing the 60 to the 250, the highlight case incorporates the labeling faster. So you can see also on the right-hand side, the slope is higher at the lower time points. And so what we can also see from pyruvate and serine, which are pyruvate's over here, and serine is part of the photorespiration pathway, which gets labeled faster than the TCA cycle. We can see that pyruvate for both cases is about the same, which means that incorporation is similar for both cases. But when we look at serine, we can see that the lower light intensity case has more labeling than the higher light intensity. And this could be because different metabolic pathways might be more active, or this could also be because in the lower light intensity, there may be less unlabeled serine compared to the 250, which is contributing to the lower labeling. And also we can see that the activity in the TCA cycle is different between the two cases. So aspartate is a proxy for acetyl-CoA, and then we also have citrate and malate. And so you can see that there is much more activity in the 250 case, which is in the solid line, compared to the 60 microeinstein case. So we predict that maybe the TCA cycle would be shown as being more active. And next we have the tag degradation, or the ACAD knockout case. So the ACAD knockout prevents tag from being degraded. And so how we can most clearly see the phenotype is when we first acclimate the cells to nitrogen replete media, and then we, at time 0, we added the nitrogen limitation media. And so we starve the cells for about a week, and then we add in the nitrogen replete media, and then we see that 24 hours later, the ACAD has much more tag accumulation compared to the wild type, which was able to break the tag down. And so when we look at the average percent enrichment data, we have malate, serine, and succinate over here. Some of the amino acids are not statistically different. So malate, serine, and succinate between the two cases is not statistically significant. However, when we look at glutamate and AKG and fumarate, we can see that there is a lot less labeling in the wild type compared to the ACAD mutant. And we predict that this might be because there is tag degradation is contributing to unlabeled carbon into these into the wild type, which is not able to be seen in the ACAD. So in conclusion, we have higher light intensity increases incorporation of carbon into the cells, which we saw at the 250 case being having a higher slope. We also saw that there is a dilution of labeling in serine that occurs in the 250 case. And the TCA metabolites are increased at the higher light intensity conditions. And then in conclusion, there are some metabolites in the TCA cycle that have decreased labeling compared to the ACAD mutant, and this could be due to unlabeled carbon coming from tag degradation entering the TCA cycle. And so future work for this project is to add this data into the metabolic flux analysis software in order to get a full flux map and a prediction of what is happening when we compare these different conditions and these different foods. Thank you very much for listening to my presentation. I look forward to taking your questions.